In the past video, we talked about ways to quantify gene flow using molecular methods if you already know which populations are which. In this video, we're going to talk about some non-a priori methods where we don't have these preconceived ideas of what populations are there, but rather we let the data tell us that information. So just to differentiate, a priori knowledge assumes discrete populations ahead of time. And yes, this is actually a term. The first time I taught this in molecular ecology, one of my students thought I just spelled prior wrong. But no, this is real, I promise, it's a real thing. So if we are using a priori methods, if we are assuming we're looking at populations off the coast of California, for instance, a priori knowledge would say, okay, we know already that this is population 1 and individuals located down here are population 2. A priori knowledge is saying we already know that. In contrast, if we're using non-a priori methods, we don't have any preconceived ideas about which populations are which, but instead we're letting the data tell us how many populations there are and where the boundaries lie. So again, if we use that same California, yes, that's supposed to be California, looks like a weird shoe, but what we're doing here in these non-a priori methods is just sampling individuals from all along the coastline. We put all of that information into the particular program that we're using, and then we let that program tell us how many populations there are. So we are not already predetermining which population is which. We're just sampling individuals from across the potential range and then looking at the data to see how many populations there are. So there are two main ways we'll talk about here in this video that you can use non-a priori methods to identify populations. Structure is one of them. So this is a particular program, and what it does is identifies the most likely number of clusters, also known as populations, in your data set. So I'll walk us through this image here. This is what a structure plot looks like. So when you run this program, this is what the data looks like. In these plots, these letter abbreviations along the bottom axis are different geographic sites where individuals were sampled from. This particular data is from octocorals that were collected in different sites in the Bahamas. So each of these letters on the bottom corresponds to a different geographic sampling site. Each one of these vertical bars represents an individual, and the height of the colored bar for that individual indicates the probability that it is assigned to, in this case, either the blue population or the yellow population. So the colors here represent different population clusters that the structure program is identifying. So if we look at on the right-hand side here, these two sites, HC and PR, most of the individuals there have a very, very high probability of being assigned to this yellow population cluster. In contrast, for all the rest of these sites here on the left, except for a few exceptions, the vast majority of those individuals are assigned to this other blue cluster. So in this case, for this structure plot, we would say that K equals 2. There are 2 clusters, and therefore we would say that there are 2 genetic populations across this whole range. Again, this differs from using prior conceptions of populations because we might have thought that each one of these different geographic sites is a separate population. But using this structure method, we're putting all of that data in and letting it tell us, and it turns out that there are really only 2 populations here, 2 different clusters. So that's an example of a really nice and clean structure plot. Sometimes it doesn't always look that nice, but there are a couple different things to point out here. This is some structure plots from my own data, and this is showing us, first of all, how you can tell structure to try and identify different numbers of clusters in your data set. And so it'll basically run through those different options. Here it went from K equals 2, 3, 4, 5, all the way to K equals 6. And then what the program does is identify, based on those different possibilities it ran, which one of those is the most likely. So if we look here, when we're adding in new population clusters, which is a new color, which is represented in structure, we're not gaining any new information, right? So for this particular data set, we're not adding any new clusters by adding a k equals 3. Everything is just kind of blended gibberish here. We're not seeing any clear structures no matter how high we go. Another thing to point out here is similar to what we've talked about before with neutral or all loci versus just outlier or adaptive loci, you can also construct structure plots based just on those different types of loci. So here in A, this is showing a structure plot, again, for different individuals from Northern and Southern California, 6 different geographic sites. If you use all of the loci, so this was the DD-RAD data with several thousand SNPs, if you use all of the data, there are really no clear population clustering here. You have a bunch of red individuals, a bunch of blue individuals, but there's no clear geographic pattern to it. In B here, this is a structure plot that was created just based on the outlier loci. So think back to that FST distribution I showed before. The outliers are the ones way out here. If you only use those outliers to make a structure plot, if you squint, you can kind of start to see some structure here. So it's definitely not as clear as that last example I showed with the corals, but in general, here on the 3 left populations, which are all Northern California, they have a slightly higher chance of being assigned to that blue cluster, and the 3 Southern California populations here on the right-hand side have a slightly higher chance of being assigned to the red cluster. So again, it's not perfect, but we can start to see some patterns, and the patterns are different if we use all loci versus just using those outlier loci that are probably outliers because they have some kind of adaptive function for having individuals from those 2 different populations survive and succeed in their unique environments. So here's a quote-unquote clicker question for us. It's in quotes because A, you don't have clickers, and B, we're not in person, but still kind of a check on your understanding. Here's an example of a structure plot from kokanee salmon. I don't know if I said that right. They spawn in 2 different habitats, stream and shore, and they sampled some individuals from a trawl sample, but they were collected during non-breeding season, so they don't know the spawning habitat for those individuals. So based on this structure plot here, we have 2 individuals, A and B. Where would those 2 individuals, where would we say they spawn based on this structure plot? So for individual A, if we look at their likelihood population assignment, has a very high chance of being assigned to the stream population. In contrast, individual B here has a very high probability of being assigned to the shore population. So this example is very similar to the assignment that I talked about with the tiger example in the previous video. So overall, these structure plots can give us information about how many clusters there are, and also if we have some individuals that are from a mixed sample, this can also help us identify where those individuals came from. And then in addition to structure plots, the other common method that's used now, especially with high throughput sequencing data, is a multivariate principal component analysis, or PCA, to identify population clusters. This is an example from cod, and these cod were collected from different geographic sites, the North Sea, Baltic Sea, and Northeast Arctic. So the idea with these PCAs is you're basically taking a bunch of different data points. So often it's SNP genotypes across thousands of SNPs across the genome if we're talking about RAD sequencing, for instance. And then what you're doing with the PCA is incorporating all of that information and then plotting each individual in a single point in 2-dimensional space. And so each individual gets its own unique location on that 2D plot in the PCA based on its unique genotypes, incorporating all of those thousands of SNPs. And the overall idea that we're looking at here is we're looking for whether individuals from the same geographic site cluster together or not. So if we look at this PCA, what we can see are 3 general clusters, and those clusters correspond to the different geographic sites. So in this case, we would say that our data is showing us that there are 3 different populations, and in this case, they happen to correspond to those 3 different geographic Cs. If we saw something in our structure plot, in our PCA that looked more like this, that would indicate there are only 2 populations because we see 2 clusters. So overall, what we're trying to identify when we're looking at PCAs to identify how many populations there are is how many clusters of data points are there.